Right. Welcome back to another session, guys. Good day to all the metric students out there. Now, we are going to continue looking at the process of translation. The process of translation. Now, as I indicated in my previous lessons, remember the process of protein synthesis involves two stages. Two stages. So, the second stage now is the process of translation. Right. Now, what is translation? To translate in the language of English, the word translate means to read the message or to read the coded message. That is translation. Now, where does translation take place? Where? So we're going to answer where and what. Where? Where does this process take place? So the places of translation, it takes place in the cytoplasm on the ribosome. So it occurs on the ribosome. On the ribosome. Now, guys, you have to be very, very careful here. We all know that the ribosome is a cell organelle, just as the nucleus, right? It's freely floating in the cytoplasm. So the cytoplasm is very, very wide because it's got so many other things that are located there in the cytoplasm. But now here we are looking at the site for protein synthesis or translation. Where site here in Dow, where does translation take place? It takes place on the ribosome. So students always talk about the cytoplasm as a site for protein synthesis. That is incorrect because the cytoplasm is very, very huge. So it's got so many things within it. So to be specific, the process of translation takes place in the ribosome. Very, very, very important. Now, let's look at this here. We have now, I will take it that this I'm drawing is the ribosome. This is my ribosome. Right. This ribosome, remember, in fact, there are so many of these structures within the cytoplasm there, right? So what happens here is that if you can remember that mRNA that was formed in the nucleus, then it left the nucleus. It comes now. Maybe we can have a look at this. We can have a look at this here. Right. Let's look at this here. Right? We have the mRNA that was formed in the nucleus there. So this mRNA now carries the coded message there and it exits the nucleus via the nuclear pore. And now this is the mRNA. It is moving. Then when it moves with the codon, remember I spoke about the codon, which is the nitrogenous base triplets on the mRNA. This mRNA, it goes and attaches itself on the ribosome. It goes and attaches itself on the ribosome. Let's take it that this is the mRNA. Right. So meaning that if we can follow that coded message, then we're going to have U, we're going to have A, G, C, G, U, and A. This is the coded message that came from the DNA. Right. So now the mRNA, it goes to attach itself on the ribosome. So this is Tumbata. This Tumbata there is what we call the ribosome. So the ribosome, as it sits on the ribosome. So the mRNA, as it sits on the ribosome, it has the codons actually exposed. Probably maybe we can actually write short notes here. So the process of translation, it goes as follows. Number one, mRNA attaches to the ribosome. This is the first point here, to the ribosome. So now with the codons exposed. So the codons are exposed, right? So what happens here is that now I have to introduce something new here. So what happens here is that having had these codon, so the tRNA now within the nucleus, we have got many types of tRNA. And these tRNA, they carry specific codes which are complementary to the mRNA. So we're going to say tRNA, tRNA as tRNA's anticodon. Remember the T's are triplets. Anticodon complements. I'll explain this one here. Complements the anticodon. It complements the codon on the mRNA. What does it actually mean here? Here we have UAC, right? So here the codon, when it actually comes and it brings, when the tRNA brings or comes to complement the mRNA, you are going to have U actually coding with A or being complementary to A. And A is going to be complementary to U. Remember, this is RNA. tRNA is RNA. So, in place of T as a complementary to A, U gets in there. A ushata no U. Ngoba ku tRNA age ku T, right? Basically, la, we are going to have C, right? And we're going to have another triplet there. Remember, they go 3 3 there. So here we're going to have G, right? Then you're going to have C. Then here you're going to have A. I guess it makes sense then. Now, as you can see that we have 3 3 3. So the triplet bases on the tRNA. These triplet bases, they are called the anticodons. And the anticodons are complements or complementary to the codons. Right. So as you can see here, I've got 2 anticodons which actually pairs up with the codons. Right. So in this case, the tRNA brings the required. So tRNA brings the required amino acids to the ribosome. So now, as you can see there now, tRNA does not bring any type of protein or amino acid. It brings only that one which is required. That is why it has to be complementary to the mRNA's codons. Very important. therefore, what happens here is that now here we are going to have one amino acid there. And here we are going to have another amino acid there. So you can see there are two amino acids there. And these amino acids are going to be joined by a bond we call the peptide bond. So we want to say the amino acids now. Amino acids are now joined. They are now joined by the peptide bond. This is very important. It's a new terminology that's being introduced here. So we've got the peptide bond. Remember, we had the hydrogen bond. The hydrogen bond is found between two DNA strands, which bonds the base pairs together. But in this case there, we are talking a bond that joins the two amino acids. So between one amino acid and another amino acid, you are going to have the peptide bond. Don't forget that we have talked about the amino acid. Amino acids, they are building blocks. So amino acids are building blocks. They are building blocks all full of proteins. So now, since we've got many proteins being joined together by the peptide bond, so therefore a protein is formed. So a long chain molecule of a protein is actually formed. And this protein now is going to be referred to as a polymer. You know, a poly means many, because it's made up of so many hundred, or one protein can consist of 300 amino acids, right? So that is now the end of the process of translation. So we have talked about two processes now. I guess you are able to understand the process. I think in my next lesson, we're going to be looking at some questions to consolidate understanding on this section. Right. Good day. Thank you.